Okay, this morning I'm going to be talking about digestion in the small intestine, and I'll look at carbohydrate, protein, lipid, and then move on to talk a little bit about the enteroendocrine cells of the duodenum that secrete two peptide hormones, secretin and cholecystokinin. I plan to move on from there to talk just a little bit about several functions of the large intestine and finish up with the defecation reflex. This will be one of many modules to come. So at least for this, it's not important that you see my face, but it is important that you see each one of these poster boards so you can use them to take notes. So when we talk about digestion in the small intestine, there are two sources of the enzymes that are used to break down macromolecules. These would be organic or carbon-containing compounds. So the enzymes come from the pancreatic juice, and we'll look at pancreatic amylase, pancreatic lipase. The pancreatic juice contains some inactive proteases that ultimately are activated in the duodenum of the small intestine, and an inactive peptidase. And I'll tell you about the difference between a protease and a peptidase a little bit later. The inactive peptidase in the pancreatic juice is activated in the duodenum. The amylase and the lipase come in with the pancreatic juice ready to catalyze reactions. They don't need to be activated like the other enzymes associated with the breakdown of proteins and peptides. So the other source of enzymes, in addition to the pancreatic juice, are the brush border enzymes. And the brush border enzymes, or BBEs, are enzymes that are literally embedded into the plasma membranes associated with the microvilli of the simple columnar cells that line the small intestine. So all of the microvilli together, as we took a look in class, form what's called the brush border, because under microscopic examination they collectively look like the bristles of a brush. And embedded in the microvilli of these columnar cells are enzymes that the columnar epithelial cells make and insert into their membranes. Brush border enzymes are important enzymes that catalyze reactions associated with digestion. So we're going to be looking at the duodenum and other sections of the small intestine associated with digestion. So just to review something that we took a look at in lab. The hepatocytes of the liver make a solution called bile. And bile flows out of the liver down a couple of hepatic ducts to a common hepatic duct, and then down the cystic duct where bile is stored in the gallbladder. The gallbladder empties bile out into ultimately the bile duct where it mixes with the pancreatic juice, and the pancreatic juice from the pancreas and the bile ultimately from the liver, but is stored in the gallbladder, flows into the duodenum of the small intestine. And that's where the pancreatic juice and the bile mixes with chyme coming in from the stomach. Now the bile contains excess cholesterol, bilirubin, and a few other chemicals. The only chemical associated with bile involved with digestion are the bile salts, which act as emulsifiers of fat. Now, the only other thing that I want to mention right now, but I'll come back to later when I build a module about the pancreas, is that the pancreas not only supplies enzymes associated with digestion, but it also supplies bicarbonate ions in the pancreatic juice. Now the pH of chyme in the stomach is very acidic because it's a concentrated solution of hydrochloric acid with a pH of 2 or lower. And all of the enzymes that operate in the small intestine have an optimal pH of around 7. In other words, they wouldn't catalyze reactions at a pH of 2 because the intestinal enzymes would be denatured at a pH of 2. So when the chyme with the pH of 2 comes into the duodenum, the chyme mixes with the bicarbonate ions in the pancreatic juice, and the bicarbonate ions bind to the hydrogen ions in chyme, removing them from solution. And as the hydrogen ion concentration goes down, the pH goes up. And the mixing of chyme with the pancreatic juice raises the pH of chyme in the duodenum to around a pH of 7, which is going to be the optimal pH at which the intestinal enzymes all operate without exception. So we'll begin with carbohydrate digestion, but I think I'm going to step back and just review carbohydrate digestion in the mouth. So, starch is a complex carbohydrate. It's a polysaccharide made up of many monomeric or monosaccharide units of glucose that are all bonded together. And when you have a long chain of monomeric units, in this case monosaccharides, all bonded together, that's referred to as a polymer, because you have repeating units, in this case, of glucose. So starch is a polymer of many glucose molecules, thousands, and also known as a polysaccharide. So starch is a complex carbohydrate made of many glucose molecules that come in from plant sources. And starch is broken down by an enzyme made by the epithelial cells of salivary glands. And this enzyme, salivary amylase, comes into the mouth by way of saliva. It operates best at a pH of 7. And that's why when this enzyme gets down into the stomach, it's denatured. So for the most part, all of its activity occurs in the mouth. And the mouth is where carbohydrate digestion begins. So salivary amylase attacks every other bond between the glucose molecules of starch and breaks starch molecules into a disaccharide called maltose. Every maltose consists of 2 glucose molecules bonded together. So if we had 1,000 starch molecules, then amylase would break that starch molecule down into 500 maltose molecules. And that's why I just put an X, because the number of maltose molecules you get depends on how much of this digestion occurs. So carbohydrate digestion begins in the mouth, and that in part helps to prevent the starch from sticking to your teeth to form what's known as plaque. Bacteria live and feed off the plaque, so the less you have of it, the better off you are. I want to also emphasize that there's an enzyme in saliva as well called lysozyme that directly attacks and kills bacteria, again to help keep the mouth sterile, because bacteria over time can contribute to dental caries or tooth decay. So now let's get down to carbohydrate digestion in the small intestine. Carbohydrate forms about 60% of our diet. And one of the most important things about carbohydrates in food is that they ultimately serve as a source of glucose or blood sugar. And glucose is the most important fuel molecule or fuel substrate used during cellular respiration to make ATP. And the ATP, of course, is essential for ATP-dependent reactions that are critical to the life of a cell. So, this is going to be a set of reactions that occur in the lumen of the small intestine. And these would be carbohydrates coming in with chyme from the stomach. So, over here I've got the simple columnar epithelial cells that the absorbable unit nutrients pass through on their way to the blood, where the hepatic portal veins carry the nutrients first to the liver before they are carried to the cells throughout the rest of the body. So, one of the carbohydrates that makes its way with chyme into the duodenum would be the starch that's not broken down in the mouth by salivary amylase. And by the way, very little starch is broken down into the mouth. 90% of the starch or more would be broken down in the small intestine. So the intestinal cells don't make amylase. Instead, the amylase that's active in the duodenum and throughout the rest of the small intestine comes from the pancreas, and as a result the amylase is called pancreatic amylase. Of course, that amylase comes in with the pancreatic juice, and its optimal pH, which is the case for all of the intestinal enzymes, whether I say so or not, is a pH of around 7. So pancreatic amylase, like salivary amylase in the mouth, breaks down starch to maltose. Maltose is a disaccharide made up of 2 glucose molecules bonded together. Maltose, as a disaccharide, is too large to pass through the epithelial cells that line the small intestine. As a result, maltose is not an absorbable unit, and it has to be further broken down to glucose molecules. So as the maltose forms from the reaction of amylase on starch, maltose is then broken down by an enzyme called maltase to individual glucose molecules. So for every maltose you would get 2 glucose molecules. The glucose would be a monosaccharide, and it is an absorbable unit. And the glucose molecules then are picked up by transporter proteins and moved into these cells. Glucose diffuses across the cell to the other side, and then transporter proteins carry glucose out of the cell into the blood, where the glucose is carried by hepatic portal veins, of course, to the liver. So, the maltase is an enzyme that is a brush border enzyme. And that means that maltase, as an enzyme, is made by the epithelial cells and found in the microvilli. So this reaction of maltose to glucose actually takes place right at the microvilli of these epithelial cells, and the glucose that results from this reaction, again, just move through the cells into the blood, where the glucose can be carried to the cells of the body and used by the cells of the body to make ATP. In addition to starch, we bring sugar into our body as sucrose. That's table sugar, and it too is a disaccharide like maltose. Disaccharides are not absorbable units. As a result, they have to be further broken down into monosaccharides, which are the absorbable units. So sucrose, or table sugar, is acted upon by a brush border enzyme. And this is an enzyme that's embedded in the microvilli of the epithelial cells. And sucrase breaks sucrose down to glucose and fructose. Glucose and fructose are monosaccharides. As a result, they are absorbable units, and the glucose and the fructose molecules move through the epithelial cells into the blood, where the hepatic portal veins take them first to the liver. So it turns out that hepatocytes in the liver take in the fructose and convert it to glucose. All the monosaccharides, ultimately, that come into the body get converted to glucose. So, in the food we eat, there are what are called sweetening agents. And they help to create the taste of food in a way that makes it more marketable. And these sweetening agents include corn syrup. Corn syrup is a mix of glucose and fructose. And glucose and fructose do not need to be digested. They're already absorbable units. So the glucose and fructose act as sweetening agents that come into our food as corn syrup, are just absorbed through the epithelial cells and into the blood. Fructose all by itself is a sweetening agent that's added to a variety of foods and beverages. And the fructose that comes in with our diet is an absorbable unit, and it too would be carried through those cells and then taken to the liver, where the fructose that comes in is converted ultimately to glucose. So there's one other carbohydrate that I wanted to talk about, and it's known as lactose. Lactose is a disaccharide, and sometimes it's called milk sugar, and that's because lactose is not only in breast milk, but it's also associated with dairy products that ultimately are made from breast milk. So dairy products include milk, cheese, yogurt. So it turns out that lactose, as a disaccharide, is broken down by a brush border enzyme called lactase. And that means that lactase is embedded in the microvilli of these epithelial cells. That's actually where the reaction takes place. So lactase breaks down lactose to two monosaccharides, glucose and another one called galactose. These are monosacs and absorbable units, and they pass through the epithelial cells into the blood, and from there into the general circulation. As galactose is taken through the liver, it's picked up by hepatocytes and converted to glucose. So this is kind of an interesting story because some people are lactose intolerant, and you may know someone like that. In fact, it could be you. So it turns out that when we're born, most kids feed off breast milk from their mom. So all kids make lactase to break down the lactose in breast milk up until the age of 4. I'm not sure why, but in some kids, the epithelial cells that line the small intestine stop making lactase and never make it again. And as a result, these kids that stop making lactase, somewhere around the age of 4, cannot break down lactose in the food that comes into their body. As a result, lactose stays in the intestine and causes cramping and diarrhea, which is pretty darn unpleasant. So the treatment for individuals who are lactose intolerant because they don't make this enzyme anymore would be, first of all, avoid dairy products. Don't drink milk, etc. But if you do want to enjoy dairy products, then you can take a lactase containing tablet before a meal, and the enzyme can help to break down the lactose so that you don't get the cramping and the diarrhea, and you can enjoy the meal. There are whole sections, generally within supermarkets like Publix or Kroger, with lactose-free foods. And a lactose intolerant person would probably pick those up. Okay, let's move on to protein digestion. And before I talk about protein digestion in the small intestine, let me just review protein digestion in the stomach. So the gastric glands consist of mucus cells, chief cells, sometimes called zygogenic, and parietal cells. And the cells of the gastric glands secrete a solution called the gastric juice. And the gastric juice flows from the gastric glands through the gastric pits and then out into the lumen of the stomach. So chief cells are cells of the gastric glands that secrete an inactive enzyme called pepsinogen, which is present in the gastric juice. Pepsinogen in the lumen of the stomach reacts with hydrochloric acid to form an active protease or active enzyme called pepsin. Of course, the hydrochloric acid comes from the parietal cells, which are associated with the gastric glands along with the chief cells. Pepsin is the active form of pepsinogen. Pepsinogen won't catalyze reactions until it's converted as it reacts with hydrochloric acid to pepsin. And this reaction cleaves off a few amino acids from pepsinogen, converting it to the active pepsin form. So pepsin is a protease, and proteases are enzymes that break large proteins down into fragments of proteins called peptide fragments. So a typical protein in kind that comes in with the food you eat from both plant and animal sources is probably around 450 amino acids. I can think of proteins that are smaller than that. I can certainly think of proteins that are thousands of amino acids in size. But generally proteins are hundreds of amino acids in size. So clearly they have to be broken down to the amino acids that make them up, because you got to get all the way down to the individual amino acids to have absorbable units. So a protease is going to break a protein down to peptide fragments. And peptide fragments typically have 2 to 15 amino acids. So often they're dipeptides or tripeptides, or maybe they have 6 or 7 amino acids, and they're referred to as peptide fragments. So it'd be like having a long string and cutting it into smaller lengths. Now, only about 10 to 15% of the protein in chyme is broken down to peptide fragments in the stomach by pepsin. By far, most of the protein isn't broken down in the stomach and instead goes without being broken down into the duodenum along with these peptide fragments. So only about 10% or so of protein is digested by pepsin in the stomach. Most of the protein, 85 to 90%, escapes pepsin digestion and simply moves along with these peptide fragments into the duodenum. So that's a review of what happens in the stomach, and now we'll go look at what happens in the small intestine with protein digestion. So protein digestion in the small intestine, and this is where most digestion occurs. And by the way, just to reemphasize something I said in class, 90% of the digestion that occurs in the small intestine. So there's a little bit of starch digestion in the mouth, a little bit of protein and lipid digestion in the stomach, but by far most of the digestion occurs with the enzymes associated with the small intestine. And most of the absorption occurs across the walls of the small intestine. By the way, absorption doesn't occur across the mouth, the pharynx, the esophagus, or the stomach. Absorption of nutrients occurs across the walls of the small intestine primarily, and a little bit across the walls of the large intestine. So the small intestine is where most of the activity associated with the digestive system occurs. Most of the absorption and digestion, upwards of 90%, occur in the intestine, and most of that in the small intestine. Okay, so protein digestion in the small intestine. The pancreas is very important to digestion. And the pancreas makes a variety of enzymes and proenzymes, or zymogens or inactive enzymes, that travel with the pancreatic juice into the lumen of the small intestine. Now, the pancreas, when it comes to enzymes, has several, and they include some inactive enzymes: trypsinogen, chymotrypsinogen, and procarboxypeptidase. These are inactive enzymes called zymogens. In other words, until they are altered, structurally altered, they can't catalyze reactions. So the proteases are really powerful enzymes that break down proteins, and if they were made and secreted in their active form in the pancreas, they would essentially do so much damage to the pancreas that it would be untenable. In other words, it can't occur. So the pancreatic cells produce these inactive proteases and then send them by way of the pancreatic juice into the lumen of the small intestine. So within the lumen of the small intestine, there is an important reaction that occurs involving an enzyme that's a brush border enzyme. In other words, it's embedded in the microvilli of those columnar epithelial cells that line the small intestine. And it's called enterokinase. This is a key reaction. So trypsinogen is converted by a brush border enzyme called enterokinase to its active form, which is known as trypsin. Trypsin is an active protease that will ultimately break down protein to peptide fragments. Trypsin, curiously enough, then acts as the enzyme that converts chymotrypsinogen to chymotrypsin, and trypsin converts procarboxypeptidase to carboxypeptidase. So trypsin forms first by the conversion of trypsinogen to trypsin. It's a reaction that's catalyzed by a brush border enzyme called enterokinase. And then trypsin converts the other two zymogens, or inactive enzymes, to their active form. So now we've got three active enzymes: trypsin, chymotrypsin, and carboxypeptidase, that form in the lumen of the small intestine. So, trypsin and chymotrypsin are proteases, and carboxypeptidase is an enzyme called a peptidase. So here's what happens. Large proteins that come in with the chyme are broken down by trypsin and chymotrypsin to peptide fragments. These peptide fragments, along with the peptide fragments coming in from the stomach, are then broken down by enzymes called peptidases. And one of those peptidases is from the pancreas. It would be the carboxypeptidase. And then the other peptidases are actually brush border enzymes known as aminopeptidases. And carboxypeptidase, ultimately from the pancreas, along with the aminopeptidases that act as brush border enzymes, break down the peptide fragments to individual amino acids, which are the absorbable units in the end of this digestive story. The amino acids, as they form, then move through epithelial cells that line the small intestine and move into the blood, where they can be circulated to the cells of the body. Okay. Let's move on to lipid digestion, and just to kind of step back and review lipid digestion as it occurs in the stomach. So TG is my abbreviation for a triglyceride, and triglyceride molecules are fats and oils. A triglyceride, also called a triacylglycerol, consists of a 3-carbon glycerol, and the 3-carbon glycerol is attached to 3 fatty acids. And the glycerol and the 3 fatty acids together make up a triglyceride molecule that we find in fats and oils. Fats generally come from animals, and oils would be extracted from the seeds of plants. So, triglycerides can be broken down in the stomach by an enzyme that's produced by the salivary glands called lingual lipase. Lingual lipase has an optimal pH of somewhere around 4, and it's active in the stomach for a relatively short period of time, but it accounts for about 10 to 15% of the breakdown of triglycerides. So lingual lipase in the stomach converts the triglycerides in chyme to 2 free fatty acids and a monoglyceride. A monoglyceride is a fatty acid attached to the glycerol. And the fatty acids and the monoglycerides that form from lingual lipase that's active in the stomach then move with chyme into the duodenum. Now, 85 to 90% of triglycerides are not broken down in the stomach, and they would just move as fully formed triglycerides with the chyme into the duodenum. So the chyme contains mostly triglycerides that haven't been digested yet, and some free fatty acids and monoglycerides from the action of lingual lipase in the stomach. So it turns out that lipid digestion in the small intestine is a little bit complicated. So I've broken it down into 2 of these poster boards. And this is going to be part 1, and the next one coming up will be part 2. So I've got a piece of tape somewhere. Yeah. Because I figured this might happen. There we go. Okay, so triglycerides come into the body as kind of globs, and you sort of know that. So, if you cut off the fat, from the side of a steak, all the triglyceride molecules that make up the fat stick together. And most people know that when you pour out an oil from some kind of plant seed like peanut oil or soybean oil or olive oil, that the oil forms a puddle of triglycerides. They're all stuck together. So fat and oils come into the body as globs. And if we looked at an individual glob, we'd see a whole bunch of triglycerides all stuck together. So it turns out that's an issue, because lipase enzymes can only operate on the triglycerides at the surface of a large glob. In other words, they don't have easy access until they literally catalyze reactions down deep into the interior of fat globs. There's only so much time for these enzymes to operate, and as a result of very little surface area at the surface of these fat globs, if there wasn't a way to break up these globs, then the lipase enzymes wouldn't be able to digest all of the fat and oils that come in with the food you eat. But that's not what happens. So the fat and oil globs in chyme are emulsified by bile salts. Now, bile salts come ultimately from the liver. Let's see. Gonna need a couple things. Be right back. Okay, I got something that I'm gonna need in just a second. So hepatocytes in the liver secrete a solution called bile, which is mostly water. There are several chemicals in bile. Excess cholesterol that the body's getting rid of because it can contribute to atherosclerotic plaque. Some bilirubin from the breakdown of hemoglobin. The excess cholesterol and bilirubin are excretory products. They're waste products that the bile helps our bodies get rid of, because the bilirubin and the excess cholesterol that goes down with the bile into the small intestine ultimately ends up in feces. So the only ingredient of bile that we care about with digestion, because it's the only ingredient of bile involved with digestion, are what are known as bile salts. I think I mentioned in class that bile salts are made from cholesterol. So they don't really look like sodium chloride. They're their chemistry is more complicated than that. So anyway, these bile salts are secreted with a solution called bile that travels from the liver ultimately to the gallbladder, where the bile is stored between meals. And then when you eat a meal, the bile in the gallbladder goes down the cystic duct and then down the bile duct into the small intestine. And that's where bile salts will combine with globs of fat and oil and emulsify the globs. Now, emulsification is a process that occurs involving bile salts as they mix with globs of fat and oil in the duodenum. And the emulsification process is going to break a big glob into tiny little droplets of fat. And that's to increase the surface area that the lipase enzymes can act upon to ensure that all the lipids that come in with your diet are fully broken down. So let me just show you a little bit about surface area. So I've got 3 erasers in my hand, and this represents the total surface area of the 3 erasers when they're stuck together. So the lipase enzymes can only act on the triglycerides at the surface, but they can't, until they've eaten through all this, get to the triglycerides deep down inside this glob. So emulsification is a process that increases total surface area because it breaks these globs up into tiny droplets. So watch what happens to surface area. If I mix this glob with bile salts, I can break it into smaller droplets. And now the lipase enzymes not only can get to all the triglycerides that I showed you originally, but now they can get to additional triglycerides and additional droplets that form as a result of emulsification. And if I do this another time, now all of a sudden the lipase enzymes can get to what we started with our big glob, but as I emulsify, they can get to, some of the interior triglycerides. And if I do this again and again and again and break up one large globule into many tiny little droplets, then the lipase enzymes have a much greater amount of surface area to attack and break down all the triglycerides. So the globs of fat are emulsified as they mix with bile salts to fat droplets. And then the fat droplets would be acted upon by lipase. Intestinal cells do not make lipase. The lipase that is active in the intestine comes from the pancreas in the pancreatic juice. So the pancreas makes a pancreatic juice that would contain those inactive protein digesting enzymes that we talked about earlier, as well as pancreatic amylase. And we also have got a lipase called pancreatic lipase. So the pancreatic juice carries pancreatic lipase from the pancreas to the lumen of the small intestine. And that's where pancreatic lipase breaks down the triglycerides in fat droplets to 2 free fatty acids and a monoglyceride. Now we're going to find that bile salts have 2 functional roles to play with digestion. So this is one functional role, that's emulsification. And now here's another functional role of bile salts, the formation of micelles. So the 2 free fatty acids and the monoglycerides that result from lipase digestion then combine with bile salts to form structures or chemical structures called micelles. Now the micelles consist of fatty acids and monoglycerides and bile salts all bonded together. And it's the micelles as they form that then diffuse through the intestinal juice to the brush border of the small intestine. And that's where I'll pick up the story in part 2 of lipid digestion in the small intestine. So this is the rest of the story about lipid digestion in the small intestine, or part 2. And it essentially begins with micelles. Of course, the micelles are formed as fat globs or emulsified and then acted upon by lipase, and the fatty acids and monoglycerides then combine with bile salts to form what are known as micelles. So the micelles, as they form, drift over to the brush border, and at the brush border the micelles literally fall apart. The bile salts are released and can be reused for emulsification and further micelle formation, and the fatty acids and the monoglycerides then enter into the epithelial cells that line the small intestine. Within these epithelial cells, the fatty acids and the monoglycerides recombine inside these cells to form triglycerides. And then the triglycerides that form inside the epithelial cells combine with lipoproteins, a special type of protein that are made by these cells. And the triglycerides and the lipoproteins complex together to form what's called a chylomicron. As the chylomicrons form, they move out of these cells. They're too big to cross capillary walls, but the permeability of lymph capillaries is greater. So the chylomicrons move into lymph vessels, which carry the chylomicrons with lymph to the subclavian veins. And then the chylomicrons are transported by way of the general circulation to adipocytes that make up the fat of the body. And the chylomicrons release the triglycerides to the fat cells where the triglycerides are stored. So we're going to move on from the digestion of carbohydrates, protein, and lipid, and finish up a discussion of the small intestine by looking at special cells in the small intestine called enteroendocrine cells. Enteroendocrine cells secrete peptide hormones, and they're found in 2 locations. So, there are enteroendocrine cells that are found in the gastric glands of the stomach, and there they secrete gastrin. There are also enteroendocrine cells that form part of the epithelial cell layer that lines the duodenum. And the enteroendocrine cells that are found in the duodenum secrete 2 hormones, cholecystokinin and secretin. So I'm going to tell you a little bit about those 2 peptide hormones secreted by the enteroendocrine cells of the duodenum. So the stimulus for the release of these 2 hormones from the endocrine cells is chyme. Chemicals in chyme entering into the duodenum stimulate these enteroendocrine cells to secrete these hormones, not into the lumen of the small intestine. The hormones are secreted into the bloodstream. And these 2 hormones, cholecystokinin and secretin, both act to promote digestive activities in the small intestine. So both of these hormones, secretin and cholecystokinin, stimulate the pancreatic cells to make and secrete a pancreatic juice that would then flow into the duodenum. And cholecystokinin alone stimulates smooth muscle cells in the wall of the gallbladder to contract. And when the smooth muscle cells in the wall of the bladder contract, they push bile out of the gallbladder into the cystic duct, to the bile duct, and from there into the duodenum. So cholecystokinin and secretin are 2 peptide hormones that are produced by enteroendocrine cells of the duodenum and secreted into the blood, and the blood carries them to the pancreas and the gallbladder, where they promote the formation of a pancreatic juice and the movement of bile with the bile salts into the duodenum of the small intestine. So I think from here, I'm just going to use chalk to talk about the large intestine. Okay, so the large intestine, I've got some notes here that I can use to sort of keep me on track. We talked about in lab. And sections of the large intestine would be the cecum. There's a little worm-like structure that extends off the cecum called the vermiform appendix. And I think I mentioned that if the wall of the vermiform appendix ruptures, in other words, splits open, then bacteria can spread from the appendix out into the abdominal pelvic cavity and set up a very active infection on the surface of those organs that can spread into the blood. And that causes appendicitis. Appendicitis can be fatal and generally is treated very aggressively with antibiotics. I also mentioned that the anus is a hole at the end of the GI tract, and there's two muscles associated with it. You've got the internal anal sphincter, which would be smooth muscle within the wall of the anal canal, and then you've got the external anal sphincter, which is a skeletal muscle around the anal canal that you can control voluntarily because it's a skeletal muscle. And that helps to control the timing of defecation. I'm going to talk a little bit about defecation in just a minute. So, as we take a look at the large intestine, let me tell you a little bit about the defecation reflex. The defecation reflex is when feces is emptied from the rectum. So, the formation of feces occurs in the rectum, and it takes about 3 to 10 hours or so for feces to accumulate and compact in the rectum before you have a bowel movement. So it turns out that as feces accumulates in the rectum, it stretches the walls of the rectum. And as the walls of the rectum stretch, it stimulates stretch receptors, which would be the dendritic ends of unipolar sensory neurons embedded within the wall of the rectum. When these stretch receptors are stimulated, they send sensory impulses into the sacral segments of the spinal cord. And within the sacral segments of the spinal cord, the sensory impulses stimulate parasympathetic motor neurons. Now my board is obviously pretty small. I have another one coming, and then I'll be able to expand the space where I can write. But I'm going to have to erase what you see up top and put parasympathetic motor neurons up top so I can finish this story. So the parasympathetic motor neurons within the sacral segments of the spinal cord, when they're stimulated by the stretching of the rectum as feces accumulates in the rectum over a several hour period, the parasympathetic motor neurons send motor impulses out their axons to two locations. One, these motor impulses through the release of acetylcholine, because this is a cholinergic effect. And again, I want to stress that the parasympathetic system promotes everything associated with digestion. There's no exceptions. And this is an example of something that is associated with the digestive process, the elimination of undigested food by defecation. So the motor impulses that travel out from the parasympathetic motor neurons that originate in the spinal cord travel out to stimulate the contraction of smooth muscle cells in the wall of the rectum. These would be smooth muscle cells in the muscularis layer. And when these smooth muscle cells in the rectum contract, when they're stimulated by the release of acetylcholine from parasympathetic neurons, the contractions push down on the feces and push it towards the anal canal. So this helps to get feces moving into the anal canal. And in general, this is when a little baby who's not potty trained yet would defecate, because this would be the defecation reflex. In order to defecate, you also have to relax the internal anal sphincter muscle. So there's one other effect, and that is to relax the internal anal sphincter muscle, which opens up the anal canal for the feces to move out of the body. So both of these effects take place in a parasympathetic response to the stretching of the rectum. You're going to push on the feces and push it into the anal canal, relax the internal anal sphincter, which is a smooth muscle that responds to the acetylcholine, and then you defecate. And again, this is when a person would defecate by way of this reflex. And it's when a little baby who hasn't been potty trained is going to defecate. We wouldn't necessarily defecate at this time because of the presence of the external anal sphincter. And the external anal sphincter is a skeletal muscle under voluntary control. So you can will it into action. And if you contract the external anal sphincter, you can hold, the feces until you get to a bathroom, at which time you would stop contracting and allow it to relax, and then the defecation reflex would take over from there. Okay, little baby, I think I mentioned in lab, generally doesn't find this muscle until it has some speaking skills and language skills. You can talk to the kid, let them know about the mother. They see that other kids around them can control the timing of defecation, and there is a moment, I guess, when they find it, learn how to control it, and of course that's going to be a prelude to getting out of diapers. Okay, so a couple functions of the large intestine. So there are a number of functions of the large intestine. We're not going to cover them all, but there are a few that I wanted to mention. And one of those is water absorption. Most of the water in the food that we eat is absorbed across the walls of the small intestine, but by far. But some of the water that isn't absorbed in the small intestine and passes with undigested food into the large intestine ends up being absorbed in the large intestine. And that helps to compact the feces in the rectum. If you literally absorb too much of that water, then the feces can get very hard, and that's a condition known as constipation. And if there is, well, not too little. That would be too much water absorption, would be constipation, where the feces becomes very hard and that makes it more difficult to poop. And if there is too little water absorption for one reason or another, and sometimes it has to do with a bacterial infection, then you end up with really, really wet feces, and that can give rise to diarrhea. I want to just talk a little bit about the bacteria that live in the intestine. So this would be a little bit on intestinal bacteria. And the numbers of these bacteria are honestly astronomical in size. So I've seen estimates of the number of cells in our body, and it's probably somewhere around 50 to 70 trillion. And the bacteria associated with our bodies that live in our mouth, armpit, in the intestine, outnumber our cells, I don't know, anywhere from 3 times as many to 10 times as many. And that means that there are hundreds of trillions of bacteria that live in the large intestine. And they account for probably somewhere around 2 to 4 pounds of your total body weight. I mean, we're really talking about a massive number of bacteria. So those bacteria, if you're healthy, don't cross the lining of the large intestine and get into the body, and as a result, they don't harm you. In fact, they have roles to play that actually help you. So most of the bacteria associated with the human body, by far, live in the large intestine. And apparently it is a great place to live. So it's warm all of the time, with the temperature around 98.6 degrees. There's plenty of food to eat, generally. That would be the undigested food, which still has nutrient value to bacteria, that comes into the large intestine. And the enzymatic activity of digestion occurs in the small intestine. And that's why that's a really tough place to live. And the high acidity of the stomach makes the stomach a really tough place for bacteria to live. In fact, the acidity of the gastric juice kills off and sterilizes, kills off bacteria and sterilizes food. So there's not all that many bacteria that live in the stomach. The small intestine is fairly hostile because of all the enzymes that would attack and break down bacteria. But when you get down to the large intestine, you don't have the acidity of chyme in the stomach. You don't have those intestinal enzymes that operate in the small intestine. And so it's a pretty, I guess, relaxed atmosphere. And bacteria seem to like it. So they got a lot of food to eat. They've got a very comfortable temperature, particularly for bacterial reproduction. They've got the water, access to water in the food that's coming in. So they got water and food, nice home, good temperature. And again, they live there by the hundreds of trillions. So our intestinal bacteria, so long as they don't get into the body, and generally they don't, are actually beneficial to us. And the relationship between our bacteria and our bodies is what's called a symbiotic mutualism. So the word symbiotic comes from symbiosis. And symbiosis is a word used to describe an intimate or close living relationship between 2 different organisms. In this case, us and our intestinal bacteria. The term mutualism means that the relationship, the symbiotic relationship, is of mutual benefit to both parties. In other words, we benefit the bacteria by giving them a good living situation where they thrive and have food to eat and can reproduce and literally populate our intestines by the trillions. And we'll talk about a few ways in which we benefit from the intestinal bacteria. So we benefit and they benefit in this close relationship, and that's why it's called a mutualism. There are other types of relationships that exist between two organisms. An example would be parasitism. So you can have a symbiotic parasitism, and that's where you have a close living arrangement between yourself and a parasite. And in the case of parasitism, one organism benefits, that's the parasite, to the detriment of the other. Because parasites can hurt you, they can even kill you. So there are other types of symbiotic relationships, like parasitism, but we're going to look at symbiotic mutualism, a close living arrangement where both organisms benefit. So it's obvious to see what the bacteria get from this relationship, but let me tell you a couple things that we get from bacteria. So for the most part, vitamins are organic compounds that the cells of our body can't make. The battery on my video camera had died just about at this point, so I'm having to retune the last segment to finish up the large intestine and splice this little segment in, so there may be, I don't know, something that's noticeable. But anyway, bacteria that live in our large intestine by the trillions have the enzymes to synthesize vitamins that our cells cannot make. And a couple vitamins come to mind. First of all, vitamins B2 and B3, sometimes referred to as energy-giving vitamins because they are necessary for reactions that occur during cellular respiration to make ATP. Another vitamin made by intestinal bacteria that's available to our bodies would be vitamin K, and that's a vitamin associated with prothrombin synthesis and clotting. So bacteria can synthesize these vitamins, and then as bacteria die, the vitamins are released into the large intestine, and they are absorbed through the cells of the large intestine that line the lumen into the blood and distributed throughout the body to the cells of our body for their use. And intestinal bacteria actually are a significant source of vitamins, and they help to supplement the vitamin supply to our body in addition to the vitamins that we get from the foods we eat. So I'm going to move away from intestinal bacteria and finish up by telling you about what's called a coliform count. A coliform count is done to test whether a body of water, like a stream or a pond or a lake, has been contaminated with feces. So there's a very common bacterium called Escherichia coli. No one ever says Escherichia. Instead, this bacterium is called E. coli. So the second part of its name, coli, comes from colon, and just indicates that this is a common bacterium that lives in the colon of animals. There's plenty of it in humans, but it's also found in squirrels and beavers and geese, raccoons. So it's a common bacterium found in the colons of the large intestines of animals, to include ourselves. So E. coli is present in feces. And if feces, essentially from sewage, gets into a body of water, then you can test for the presence of E. coli. And if you find E. coli, the source has to be feces of animals. And that means that the body of water was contaminated with feces. And as a result, it would be unsanitary, or certainly not safe for human consumption. And you wouldn't want to swim in water that was contaminated with feces, as indicated by the presence of E. coli. So there's a test for the presence of E. coli in a water sample that you take from maybe a beach along the shores of Lake Lanier called a coliform count. and you bring a water sample, let's say from the beach at Lake Lanier Islands or some other beach, into a lab setting, and then you test that water sample for the presence of E. coli. If you have a little bit of E. coli beneath some threshold value, then the water's clean and it's okay. You know, who knows, maybe it was a couple geese or something like that were swimming around, and no big deal. But if there is a certain level of E. coli contamination to your water sample, they'll close the beach down, and they'll continue to sample until the E. coli count, as indicated by a coliform count test, reaches a point where, the water is safe. So I do want you to know the name of this test for fecal contamination of water sources like ponds and streams and lakes and beaches, called a coliform count. And I think that concludes everything that I was going to talk about with respect to the small intestine and the large intestine. And of course, I'm appending all of this to what I've already covered over the digestive system. I'm going to create another module to tell you about the pancreas and the liver. And the pancreas and the liver will finish up what I want to tell you about the digestive system. Although I think at the end, I'll tell you about a couple eating disorders and some miscellaneous topics as well.